We are entering into another topic of this morning, and I'm glad we're all getting refreshments and food and sugar, so hopefully you'll be more engaged for the next session. So we're calling this HCA Asia Flagship Project, and this is the first time we're trying this out. And the reason why we thought it would be a good opportunity is through the success of what happened through AIDA project led by Sham and others. We thought, why can't we do another wonderful initiate a new flagship project here in Asia? So we invited few speakers who are an expert in the fields of cancer, infectious disease, microbiome or gut biology, full-length cDNA technology, and single-cell GTEx or EQTL. And we just kind of asked them to give very brief pitch about what could potentially be done in the region. So the main message here is that you have to pitch something that cannot be done in your own lab or cannot be done in your own country, but it really requires a regional collaboration. And I think these topics like cancer and microbiome really requires a collaborative effort throughout this region. And I hope this will create the new rounds of projects like AIDA that can not only help to bring people together, but creates a seed for training and workshops that are built within these programs and bring the equity goals in the process. So we're really excited, and I will really encourage the speakers to keep within time because we also want to have a lot of discussion around this. So first, I'm going to invite Woon-Yang and his team. And because of the interest of time, I'm just going to ask the speaker to just give a self-introduction, your name and where you're from, and take it away. So these chairs are for later. Yes.
Okay. I'm Young Park from Samsung Medical Center. So I invite our collaborator for the introduction of the Asian Cancer Atlas project. So in that initiating, I'm not starting, but actually we propose that as a kind of HCA Asia collaboration project. So can I see my slide? Sorry. Okay. So we have the four speakers today. So the reason why I propose that Cancer Atlas to be as known, some difference in the clinical response to the immunotherapy. Also in that some demographic difference in the cancer, as you know, is that there's so many reasons for the some environmental, also genetic the background. So I will present one case, and the other three speakers also present their cases. So when I analyze that colon cancer, compared with our data against the other Caucasian data, so the pattern looks like same, but actually so markers are also same, but actually we do see some difference in the population in the TME. So TNK is high in the Asian population versus the B cells low in the Asian population. Also critically actually the pattern for the T cell subtype in colon adenocarcinoma might be different in the Asian population, more exhausted T cells, and versus the white people, more the memory cells. The breast cancer also, they show the pattern like the more exhausted. So when we experience that kind of immunotherapy response is higher in the Asian population, we can understand that this kind of difference may explain that reason. Also, maybe we can accommodate more in the clinical trial, also drug development for those kind of knowledges. So, and then the teeny.
Okay, so 1 minute. Okay, I'm Teeny Jinawat from Thailand. So just want to show you that we have this pilot study of colon cancer ascites of the Thai patients before, and we hope that in order to develop all this pipeline for tumor biobank-based systematic clinical specimen sharings and a multi-center cohort, we have tried to optimize the protocol for cryopreserved single-cell dissociation from fresh tumor tissue, and also for a protocol for single nuclei dissociations from the archived biobank tissue. So right now, we are quite ready for all these collaborations, and we are happy to do so if you have any interest in that.
I'm Ankur, and HCC is, which is primary form of liver cancer, is a historically Asian problem, and now we are seeing more evidence of because of metabolic disorder, HCC emerging in Caucasian countries. So the two places where I have worked, and when I have worked on HCC data, I analyzed these two data sets. One is coming from Singapore, majority of ethnicity is Asian, and another is coming from Perth in Australia, which is assuming most of the patients were Caucasian. And we analyzed these two data sets from HCC tissue and looking at lymphoid cells. We could see that there's some enrichment of T cell population within, which looks like regional specific. Again, we need to generate more data, but it appears that the T factor cell, which are the main target of immunotherapy, is differentially distributed in at least in these two cohorts we have studied so far. So for understand this into more detail and to understand how immunotherapy might be impacting different ethnicities, we are conducting this project where we are collecting samples from London, Singapore, Seoul, Perth, and Sydney. And we're looking into these patients who are getting all our advanced stage HCC patients, so one type of HCC, and all of these patients are getting same immunotherapy, Adzo plus Peva. And we are collecting blood at the same time points. So this will allow us to understand how ethnicity, Caucasian versus Asian, might be impacting in terms of etiology, viral versus non-viral immunotherapy response. And yeah.
So very quick. Yeah, here. So my name is Suzuki from the University of Tokyo. I'm not representing the entire Japanese team, but I'm at least in charge of the sequencing collaborative at the University of Tokyo. Perhaps the important thing is that the 10x Xenium and the BZI Stomics, our latest platforms for the spatial analysis, are at the final stage of the setup, thanks to the dedicated efforts of the 10x Genomics and the BGI technical staffs. And Brett and Wei Bin somewhere are in the hall, so please catch them if you have any further technical questions. So we want to leverage these platforms for the analysis of the cancers, Asian cancers, that community-wide. So the targets will cover not only, but not restricted to, the usual cancers in terms of the Asian-specific, unique characteristics, features of those cancers are for example, lung cancers and colorectal cancers, and also as well as the regional cancers, which are unique to this region. For example, the adult T-cell leukemia is distributed through Asia, almost exclusively in Japan, and together with some unique anti-cancer drugs. The volumetric study is just approved in Japan and under review in FDA. And we have already observed to see the sign of the relapse, and the single cell analysis is definitely needed to understand the relapse mechanism of this unique cancer after the treatment of these unique drugs. And we look forward to any proposals for the collaborations.
So we discussed about some potential collaboration project under the HCA Asia. So the goal would be we can tackle with some problem and clinical issues, but also we can provide some comprehensive map for the Asian cancer cell. So maybe mostly the immune cell will be interested about, but actually we also want to incorporate some platform for cancer single cell research for any countries in the Asia. Also, we would like to provide a database for the clinical and genomic data together for the study. Because as you know, the cancer cell single cell data is not. I mean, they maybe provide many informations, but also we need the clinical data is more critical to understand that those single cell data. So we would like to cover that many technical and also many the part. And then, but initially we can start without funding. I mean, for example, we can aggregate data by ourselves, and then maybe we can collect more samples from the inside of the Asian countries. Also, we're gonna share the protocol and harmonize everything and also starting to develop the database first. And then in the perspective stage, with some new data, I mean, we need some the support from the outside, and then probably then we can generate more data, and also we can expand those kind of study together with under HCA Asia. Thank you.
Thank you. Maybe if Woon-Yang can stay up or what, yeah. Thanks for keeping in time. So we actually have 2 minutes for just Q&A, or if anyone want to ask questions to them. So I actually have a question to Unyang. So I think what's the strategy in terms of which cancer to start? Because obviously it's very heterogeneous, very diverse. Yet there's potential to really focus on one cancer, or do you plan to just collect everything that's available?
It's actually based on some difference in the against the Caucasian population. For example, in the colon cancer, we have some difference. Also in some environmental diet in the different in the some Western versus East Asian. In Asian, definitely we can find some difference. Also, liver cancer be a problem in the Asian countries. So I think maybe second. The third be a lung or maybe other type of cancers. That based on the number of data, also based on the difference, right? Proved. Yeah.
So it's kind of prioritizing based on gut, liver, and then lung would be. Yeah. That's a good idea.
Shao.
Wang, it's very exciting. I'm reminded of our early discussions before we planned IDA, where we were very worried about batch effects between different countries.
Right.
So how would we handle that in this, not just in this consortium, but in all of this flagship project discussions we're going to have today?
Right. So we learn up from the some IDA. I think maybe we need to analyze all the protocol first, but actually at this moment, we build up the protocol first for the cancer atlas. But starting with the previous data, we need to develop the method to how can aggregate the data together to understand that any difference between those populations. So we definitely need a new protocol to harmonize all the data. Right.
Thank you.
Maybe one quick question there.
I'm really glad that you talked about lung cancer, because I think from what in our initial analysis, it seems like Asians have a very unique mutational signature pattern associated with open smoke. So yeah, I'm really glad.
Yeah, definitely lung cancer be a good candidate for, because that we have the high EGFR mutation. Also, the immunotherapy response also very nice in that, right?
Quick question.
Just have a quick comment, which is that if you're going to include multiple types of cancer, I think we need to pay attention to the epidemiology, the prevalence and incidence of the various cancers as well.
Right,
Thank you.
definitely. Yeah, thank you.
All right, thank you very much. If you can have the next speakers for infectious disease, Fardom and Yuki. So I'll have you guys again self-introduce yourself and where you're from, please.
Hi, I'm Waradon Sangnak. I'm a principal investigator at Mahidol University, Thailand.
Hi, this is Yuki Norakata from Riken Institute and Osaka University, Japan.
So for our session, in terms of our attempt for try to pitch some ideas, so it's more on trying to find consensus with respect to infectious disease. We think one converging point that could be a great starting point for collaboration would be COVID-19. And that's because in terms of infectious disease, the data that we have been collecting regarding like any infectious disease at all, in terms of COVID, this has been unprecedented because the data that have been generated have been tremendous, and we've learned a lot from this single infectious disease, ranging from human genetics, cellular phenotype, and responses from different demographic groups, different clinical outcomes, different clinical measurements. So I think the idea that we can try to collaborate more can be integration from both horizontally, right? So expanding the groups that also include samples from maybe underrepresented groups, like underrepresented country, and at the same time we might try to encourage more integration vertically. So including data ranging from like the host site, genetics, cellular heterogeneity, responses, different outcomes, and also from the pathogen side, right? Different strains of the pathogens, and all together, right? Like we can expand opportunities that we can like study this problem in a really, really comprehensive fashion.
Yes, I have some additional comments. So this working group is trying to focus on the infectious disease, and as we have seen these sessions, so infectious disease is a good target of single-cell mixed resources. So we should definitely start focusing on some specific disease and make a collaboration network using agents. And also another point is the infection status is also a kind of a strong stimulation status of the human immune system. So in some sense, focusing on some specific infection disease tells us how the immune system changes when we have strong stimulation. So we are now trying to find which disease is good for the collaboration. Of course, COVID-19 is good, and maybe some of us, we already have some data. So collecting these COVID-19 results together is one of the good recent targets. But I think COVID-19 pandemic anyway is now going to be shifting. So we definitely need to change another type. If there comes another new pandemic, it is of course a good target for us. But also we should also think as still existing but clinically problematic diseases such as tuberculosis or more population-specific infectious diseases. And maybe here's a good partnership among Asian countries. So we are definitely happy for collaboration and coordination with all of you guys to find a single-cell sequencing for the infectious diseases.
Great, thank you. Thank you very much. They're open for questions, but maybe I'll start. So you mentioned COVID-19, but we've seen presentations on dengue, and there are some speakers who work on malaria. Any thoughts on tropical-related infectious disease?
Yeah, so I think this is a great opportunity to explore other like tropical or neglected infectious disease as well. And I think at least in Thailand, I suppose there are lots of microbiologists here who already have been collecting samples to study pathogens. But now, right, like we bring together like scientists who also look at like the cell side. So this could be an opportunity that we could collaborate and then utilize the sample, right? So coordinating with scientists who already collect like samples from diseases that look at the pathogen side, and then having a collaboration for scientists who would look at the host side. And then, like collecting variants from the pathogen, and then on the host side, we can get information from the immune responses or other like cellular host responses that might allow us to understand or get information in a much, much more comprehensive fashion.
Yeah, I think, previous cancer topic, I think defining potentially which area you want to target that can kind of open up avenues for key leaders in these countries in the region to kind of raise their hands, then come together. So I think this could be maybe a common theme for today, is kind of clearly defining perhaps which area that you want to pursue or target may also help to reach out to the right people for collaboration as well.
Yeah, I also think that infectious disease with different clade has different host genetics involvement, different clinical symptoms is a good target. So it means that even though the same infectious disease like tuberculosis, the majority of the clades are different among regions and countries. They may have some different types of symptoms. So it's even though they are prevalent disease, a good target for collaboration.
Yeah, excellent.
Yeah.
So you mentioned COVID-19. I think most of the talks that we're talking about single-cell work we're focusing on peripheral mononuclear cells, right? But patients die because the lungs stop working, and there are very few studies on actually the lung samples. So is there some thought around those sampling that aspect and generating data from there?
Yeah, I agree. So maybe we focus on infectious disease. Which kind of tissues other than blood we should collect is a good point of discussion. So maybe blood is a basic tissue, but also, you know, the lung, the valve or lung tissue is also strong target for the lung infectious disease. So I'm not sure every country can collect this due to some restriction of the infection boundaries, but I totally agree that this is an important topic.
I think, again, there's an opportunity where we can also connect with the lung bionetwork of Human Cell Atlas and kind of train people on how to collect samples and profile them. So
yeah,
it's an opportunity also to connect with the global HCA community. Christine? Yeah.
So I think this is an incredibly important atlas for a couple of reasons. And arguably, you can't interpret the behaviour of an immune cell in the absence of some kind of immune challenge, because that's what they're for, right? But of course, when you're thinking about infectious disease, there are a couple of facets that you need to consider. The first is what has gone before, so what have your donors been exposed to already? How does that influence the disease you're studying now? And what are the consequences in terms of the pathology and outcome? And I just wondered how you're thinking about those facets in the construction of this atlas.
Yeah, I totally agree. Yes, I definitely agree to your suggestion. Yeah, I think. Yeah.
So starting with what's gone before, how will you account for the primed status or the memory status of lymphocytes and B cells to an infection that someone might have been exposed to in childhood?
I'm sorry, can you explain that?
So like metadata and the experiences, the previous exposures to these viruses.
Well, obviously you capture it through metadata, but I would expect that just as you see a smoking signature in lung cancer, you would see prior infectious signatures in people where there are some diseases endemic in that region, but where most of the population are exposed early in childhood, which may actually have a profound influence on how that population responds to later challenges.
I agree. So exposure itself has a strong, in some ways, bias for the status. And I think also this is strongly always a discussion for which kind of control we should put, healthy or exposed but no symptom. I think, anyway, I agree, metadata including, I think, severity or mostly severity or outcome is an important outcome clinical information when we do the infectious disease analysis. So, because sometimes it can, even though they are exposed, this difference in the outcome can differentiate the biological status and signature of the infection status. But I agree, which kind of clinical metadata we should collect is also important topic.
Okay, sorry, for the interest of time, we have to move on. So I know there are a few interesting questions. And again, this is a very important topic for many of us. So please come reach out to those speakers.
Yeah, please freely contact us, any kind of collaboration. We should definitely make discussions.
Okay, so thank you. I'm going to ask Arshita to come up and give us your talk about microbiome, please.
Very good morning to all of you. And thank you to the organizers for giving me the opportunity. And a big shout out to Thailand. Wonderful place, beautiful people. I'm Archita. I'm from Perth. I'm a new PI in Telethon Kids Institute. And my major interest is to understand how the microbiome evolves in early life and how it affects the development of the immune system. And talking about microbiome and people, we all know that humans are a constantly evolving ecosystem where we are partly human and partly microbial. If we really have to count the number of genes that is encoded by our microbiome, it's 100 times more than, I hope everyone can hear me. It's 100 times more than what our genome encodes. And we have like just 100 trillions of microbes just present in our gut. This is one organ. So they are very important. And we as human have evolved as holobiont. And we have to understand that in order to study human physiology, we cannot exclude this very important organ, which is microbiome, as part of our whole analysis. And yes, people have tried to do it in the past, and they have been successful as well. In 2007, the Human Microbiome Project came into picture, and it was a massive effort to basically understand what kind of microbiome is present in humans. And the problem with that entire effort, which was quite successful, but at that time, like other fields as well, it was considered to be a static entity where we have microbes present in our body and they just wanted to profile it. But today, like many other fields, we have seen that everything in our body is dynamic, and so is the microbiome. It changes with age, it changes with what you eat, with region, demography, geography, and all that is just one layer. The another layer is the difference between the commensal and the pathogenic organism, which today is kind of blurry. And I think it's going to stay like that for long because there's no such thing like good or bad bacteria. It's the state in which we are that defines how we are going to respond towards these microbes. But nonetheless, the microbes are an important part of our physiology. They are intertwined right starting from the development till the time we age and we become old. It's still important for our physiological functioning and the way we encounter diseases in our surrounding. And I was very fascinated by what Jason showed yesterday about the periodic table of human atlas, human cells, because I have been thinking about the periodic table of microbes in our body since long, and not just our body. It's basically the microbes which are present in the surrounding. But we can start with the body and with the organs in our body and see what kind of microbes are present there and how we can tabulate them, not just in terms of the organs that we have, but also in terms of how it changes with time and how it shapes our immunity, which is the most important aspect. And just one anecdote here that why we have not totally understood how we have evolved with the microbiome is because the microbiome has not been found in most of the fossils so far. So we don't understand how the evolutionary process during the microbial diversity has changed. And the only evidence we have in the fossil record is whenever the microbiome has been fossilized with dental plaque, which was not a lot. So we really don't know how our ancestral microbiome looked like, but it will be a good start to know how it looks during the development of a single individual and then how it changes with time. And with that, I want to propose that we should now make an effort to understand the diversity in the microbiome, not just the microbiome present in our body. And to understand that, we have to start with specific regions which are unique in their identities and the way they function. And I propose to basically sequence the microbiome of Asia-Pacific region. I'm coming from Australia, and we have amazing collaborators present in different other countries like Singapore, Malaysia, India, Australia. And I really encourage more people from here to come and talk to me, and let's join hands to study the microbiome present in our body across regions. And when I talk about this, we have to understand that the world is changing. And we have seen it in the past few years very closely through COVID. But even before that, the world is changing, people are changing, and we all are moving constantly. And that has a huge impact on the way our microbiome is shaping these days. And that's why I propose that when we talk about the microbiome, yes, we are talking about Asia Pacific, but eventually it all has to come together, and we have to understand the microbiome during migration. And that will tell us how the microbial diversity changes with place and how it can impact our well-being. Because every place you go will have a different kind of allergens and pathogens, and that impacts a lot the way the immigrants especially survive in a different country. And it's an interesting thing to understand how the immigrant microbiome changes. And another layer, like just thinking ahead of time, is to understand how the first generation and second generation immigration changes. Because a lot changes from first generation to second generation immigrants, the way they shape their life and the kind of cultural diversity they encounter with time. So that is another important part of understanding the microbiome. And sex-specific changes, definitely, because we know how different men and women are, and the longevity issues and all that, the kind of diseases which are associated with certain sex. So it is important to include that part also here. But all in all, to start talking about the diversity in microbiome and start sequencing it and start understanding it at single cell level at different times of life. And for that, I propose that we cannot, again, as we cannot study the human system in isolation, we should not also study the microbiome in isolation. And that's why the whole idea and approach I want to propose, and I want your opinion as well, is to sequence the microbiome, and at the same time, if possible, and I think it would be possible because it's very important, to also profile whatever human immune cells we can profile together. And that will give us a parallel view of how our immune system is changing or how it is shaping the microbiome or vice versa. So I propose to collect fecal samples and oral swabs, and then we can start with metagenomics so we can understand what these microbes are doing there as well at whole genome level. And we also should do culture-based sequencing because, of course, like that will tell us better about the kind of conditions these microbes survive in and eventually how that's going to impact the human body, and the single cell profiling of the human immune system. And at the end, like what is important is to also try to study them together. And for that, so we have already recently published a work where we have shown that human fetal organs have microbiome, and it kind of has a memory, the immune memory in the fetal tissues, and it shapes the immunity in a certain way. So the baby is ready for the external exposure later in life. And this particular thing tells us, and various other evidences as well, tells us that our development is not just about our human cells. It's about all the other forces which are coming together to shape the development of a human body. And same as with any animal, but I'm just talking here about human. And for that, we have to study this in unison. And that's why I also propose that when we are doing this, we should have a massive collaborative effort with as many people as can be involved, and also study the microbiome and immune system together. And of course, these kind of studies always are haunted with contamination issues. So we have developed a very unique and very comprehensive pipeline to study the microbiome and just remove most of the, or like 99% of the contaminant issues which can occur in such studies. And this is the pipeline for that. And I would love to discuss later if anybody wants. But again, as I said, the microbial control of immunity is very important. And for that, I want more and more people to come together, and whatever kind of samples we can get in this space. Here I propose that if we get human biopsies, if we get the FFP blocks of human biopsies, which are already present in a lot of pathology laboratories, and we are already started to collect them across countries, and then we study the immune system through spatial transcriptomics and the microbial system through imaging, and then combine them together to study how they interact with each other. And we have already started this effort as well. We are currently using nanostring technology for the spatial transcriptomics, and we are using RNA scope for a bacterial imaging, and now we are trying to study them in unison together. So as I said, like the kind of study I want to propose is Human Microbiome Diversity Atlas. And the important thing is, like Sarah mentioned yesterday, that mechanistic impact and clinical significance is very important. And I think we won't be able to get there unless we really understand all the forces which are present there along with the human body. And microbiome is a very, very important part of that entire system through which the humans evolve and the way they function. And at the end, because I'm talking about this from multiple perspectives, we study the microbiome and the immune system, it will be very important to integrate it with other successful stories like IDA, which Sham mentioned yesterday, and to combine them together, and then we'll have a better picture to understand the diseases of certain regions and how we can be more clinically relevant in disease space. And with that, I would like to conclude and thank everyone, and let's join hands and please come and talk to me. I really, really appreciate if anyone will come and talk to me about this later. Thank you.
That's fantastic. Any questions? Maybe I think spatial transcriptomics is going to really open up an opportunity here where you can combine human cell atlas with the microbiome. And I think there's a growing technology where you can actually profile S16 in conjunction with spatial transcriptomics.
Yeah.
So I think it's going to be a fantastic way and opportunity for technology, but also building human cell atlas and microbiome at the same time. So.
Yeah, that's correct. We are talking to BGI about that, and they are actually trying to develop something similar where you can profile the entire microbiome and not just 16S, but basically the whole genome. Yes. So that will be very exciting to take on in the next few years.
The kind of multi-omic strategy.
Can I make a comment? Yes. One major challenge in this project will be to look at, understand which tissue to look at these immune activation, etc. Also, the microbes will be so diverse that it may not be easy to correlate unless you're looking at the dominant microbe.
Yeah.
So, but anyway, these are matters for discussion.
Yeah, I agree. I agree with that. And that's why I think a good starting point will be to understand what kind of samples we can get on the global level. And that will be the stool samples are something which we can easily access. So good starting point will be gut and to understand the gut diversity, because that changes very frequently with our move or the kind of food we are eating and the kind of antibiotics we are taking. And yeah, eventually we need to move much closer and understand how it's changing in other organs and other diseases as well, like cancer and infectious diseases.
Yeah, great.
Yeah.
You mentioned gut, so Unyang mentioned colon cancer, so something to think about.
Yeah, okay. we were talking about that last
Excellent.
night.
All right, thank you.
Yeah. Thank you so much.
So I'm going to invite Alistair Forrest, and he'll talk about ways to utilize some of our 10x cDNA libraries. So looking forward to it. Thanks.
Sawadee krap, and g'day, I'm from Australia, my name's Al. So this is really just a pitch. I think now is the time to actually be looking at doing single cell full length much more broadly. Disclaimer, I have some content from ONT and from PacBio and obviously 10x as well. It doesn't mean I endorse any of these products, but they're there. Okay, so this is an image actually from PacBio, and the concept is you're familiar with the UMAP, you've got all of these different cell types you've identified, but what full-length transcripts are actually, what isoforms are present there? Are there other genes that you haven't actually got a transcript for in your reference transcript database? So why? Full-length catalogues are critical to understand the output of the genome. The first thing that we do with our short-read libraries is we map them to transcript models. And then anything that doesn't map to it, we throw it away, typically. But the cool thing is, full-length cDNA has generated almost all of these protocols. So you guys probably have got full-length cDNA sitting in your freezers. So it's not going to be extra cost to you, it's just sitting there waiting for someone to help make that into a full-length compatible library for sequencing. So this is from John Bolaurier from ONT, and it's just summarising the structure of the 10x libraries before they're made into short-read libraries. And basically the main point there is that there's the blue bit in the middle, there's a full-length transcript there. So these can be converted into libraries that are compatible for sequencing on Nanopore or on PacBio. So I think there's an opportunity to extend gene catalogues using full-length cDNAs from rare cell types. And just to give you a sort of background on why you might want to do this. So many of you would know we've been involved in Phantom for donkey's years. But in Phantom 5, we did bulk RNA cage profiling of primary cells, cell lines and tissues. And at that point, there were about 1,200 protein coding genes we just didn't see anything for. If you went and looked at the literature on each of these, the vast majority were expressed in rare cell types. So the catalogues that we're using to map our short reads to are incomplete. So there's a potential for us to actually discover new transcripts, discover new genes, if we were able to generate full length from these rarer cell types. And the reason I'm talking about rarer cell types is typically the catalogues have been generated. Here's a lump of heart, here's a lump of kidney, here's a lump of ... or here's a purified population from PBMCs, but it's not going into these really, really rare cell types because the technology just wasn't there yet. And now there are kits available. Oxford Nanopore and PacBio both have products. So I'm going to try that. No, that's not, okay, I won't. So the path at the top is what you're all doing at the moment. You make a full-length cDNA, it goes into a short-read library prep, and then you sequence on some sort of short-read sequencer, typically an Illumina. But those full-length cDNAs can be made into a long-read library, sequenced on ONT, or this is the PacBio version where they're concatenating it all together. So there's kits available right now. I'm saying I've got 10x there as an example, but you could do it with single run, you could do it with pass theoretically. It should be agnostic to the technology that we're using. Okay, so the capacity is there now. How would we actually roll this out as a platform? So you can do the brute force. So basically, you take your library and you sequence it deeply. The problem with that is most reads will be from abundant cell types and also abundant transcripts. You could deplete abundant transcripts, so you could use like CRISPR depletion, or you could perhaps develop some sort of subtraction protocol. What we've played around with a little bit, and this is really old preliminary data, try to do rare cell enrichment and basically use the short read information to tell you which barcodes are worthwhile targeting, which barcodes belong to these rare cell types. We use PCR. You could use a pull down with a biotinylated probe. I'll skip over that, but basically we're doing a PCR with one of the primers targeted against the barcode of the cell that we care about. This is a mouse kidney library that we published on a few years back, and the arrow is just showing some podocytes. So podocytes are really important cells of the kidney. It's fairly rare, so we were able to amplify up these podocyte-specific transcripts and generate full-length cDNAs on a minion at that point. So that's one podocyte-specific gene, podocin. This is showing detection of alternative promoters. We also see alternative splicing. Okay, so you could do brute force, you could do targeted. How would we do this as a collaboration? Why am I talking to you guys about this? So sort of historically in the region, there's experience in doing full-length annotation projects. There's lots and lots of single-cell libraries in your freezers at the moment, and you will have some interesting cell types that haven't been deeply profiled, that haven't got full-length cDNAs generated yet. So for you to contribute, it's a minor thing. It's a tiny, you've already got this library sitting there. You just need to generate it, convert it into a full-length library and sequence it. And obviously you need the money to do it. So the data generation, you could have a hub model where collaborators send some of this first-strand cDNA to a central location. That's converted into a long-read library. It's sequenced. You could have a local model where the labs generate the full-length data themselves, or they generate the full-length libraries, and then it's shipped somewhere for sequencing. Analytics would have to have a standardised QC workflow, complete versus incomplete, etc. We want to stick this all into a genome browser, and we could distribute the annotations like we've done previously in the Phantom projects. How would the framework work? A collaborative framework work? It would be open to anyone. If you contribute, you got co-authorship. There'd be a contribution statement for all authors. We need to get funding for the libraries and the sequencing, and I think I'm up. And acknowledgements to Matt Jones and Dahi Murray for generating the data, and Caleb Thomas for doing the analytics. And please feel free to drop me an email about this.
Thank you. It's such a great it's an opportunity that we can really tap into, and it's really exciting. And you showed even spatial could be considered.
Yeah. So we've actually tried some Visium full length. Actually, we're trying to do P53 genotyping. It didn't work, but we'll get there eventually.
Awesome. Yes.
So it's finally really great to see long read make it to the grand stage. As I remember, one of the first grants that I submitted for HCA did not go through, but maybe now is the time. I think there will be two challenges, and we can talk about it more. One challenge is the library prep itself, because typically the workflows that you have from 10x, they tend to have more PCR cycles, which, especially for long reads, biases towards more shorter ones. And we can talk more about it. And the other big challenge is the data analysis itself, not only just picking out the cell types and mapping them back to long reads, basically mapping between short and long reads, but also exactly how do you actually annotate what you've got. Because unlike a bulk experiment where you'll have a lot of depth, there is typically a lack of depth with the number of molecules that you'll be getting, and there's going to be a lot of molecules lost. So really happy. I've been happier if the 10x pipeline that you showed had the reference for my paper as well, which came out in 2018. But yeah, really glad.
Okay. All right. Well, thanks once again. Yeah, for the interest of time, we have to move on. Thank you, Sean. So for the last part of this flagship project session, we're going to invite Sham back to the stage, where he'll talk about single-cell GTEx, or an opportunity to do EQTL across different tissue types. So Sham, please.
Thanks, Jay. This is an amazing session, and so excited about all the flagship projects everyone has proposed before me. And I'm really hoping, so before I talk about this, I'm really hoping that when we leave Bangkok already with some teams formed around these ideas, and then people are already writing up the project plan on the flight back home, right? Or for people in Thailand on the train or car back home. So that's really the goal of Human Cell Atlas Asia meetings, to seed these kinds of collaborations. It's also to disseminate science, to get to know each other, but the output has to be, there have to be some collaborative projects across HCA Asia. All right, I'm going to propose one more over here. Just 2 slides, and then looking forward to discussion. And I hope in the break, people will come up and talk, and maybe we can nucleate a new idea, right, for a new flagship project. So you all heard about GTEx, and if you haven't, it's a consortium, at least co-led by Christian Ardley, very impactful, doing bulk gene expression profiling across 50 or 100, 500,000 individuals. I think primarily from cadavers, and then genotyping them as well, and identifying expression quantitative trait loci. Okay. So genetic variants that correlate with gene expression, and presumably some of those genetic variants, they're in LD with, or they're actually causal for that gene expression variation across a population, right? And it's a very powerful tool in human genetics, but you can all being at a single cell meeting, you can all immediately appreciate that the key limitation is that that's based on bulk RNA-seq, bulk sample analysis, right? So the obvious next thing to do is single-cell GTEx, right? So marry the philosophy of GTEx with single-cell technology, and then do the same thing and do single-cell expression profiling in large cohorts, genotype them, and find single-cell eQTLs and single-cell splicing QTLs, and with the long-read technology, for example, that Al just introduced or described. So this is very exciting. This is a real frontier, I would say. And again, Jay and me and a few others showed these images of the existing genetic diversity or ancestral diversity in GTEx, okay? And it's, as we mentioned, it's largely European ancestry in GTEx right now because of the way it was funded and the geographical constraints of the original cohort and so on. But of course, the GTEx consortium are acutely aware, being geneticists, they're acutely aware of the need for diversity in their cohorts. And so now in this second round, in the single-cell GTEx, they're putting diversity right in the study design front and center and the project planning, in the consortium planning, right? And not just ancestral diversity in a small number of countries, but even geographical diversity in recruiting cohorts from all over the world. I mean, they're coming to meet us, and so we have to join them, right? We have to help them achieve their goals and our goals of doing diverse science and profiling the whole human population. This is really our chance. So I'm hoping that Human Cell Atlas Asia, people here and your colleagues and friends will come together and propose some tissues where you have access to, so let me come to the logistics, okay? So what do we want? We want Asia-based cohorts where we can do single-cell profiling and genotyping and identify QTLs. And once you have single-cell data, you can identify 100 types of QTLs, right? They're not just plain vanilla genotype with expression counts. So you can correlate genotype with abundance in expression space in some very narrowly defined slice of gene expression space. You can correlate it with, say, trajectories of response to some stimulus, again, in expression space. So you can correlate it with noise, the noise level in expression rather than the average expression. There's really no limit to the kinds of QTL analyses you can do. And all of those, I think we heard about response expression QTLs yesterday being genetically very relevant. Chung was talking about QTLs and connecting them to chromatin state and saying that that's a very nice way of prioritizing GWAS variants. So scientifically it's hugely exciting and aligns very well. So the goals of this new consortium will align very well with the goals of the Human Cell Atlas Asia. Fantastic opportunity. What we need is to narrow it down to a small number of cohorts. Obviously, they cannot do 100 different tissue types. It'll be a handful of tissue types, because if you want to profile cohorts all over the world, then that's a lot of money, there's a lot of samples, a lot of effort. you cannot do every single sample type. Right? You have to compromise somewhere. So what are our priority sample types in Asia that we feel is relevant for our local science and that we have access to tissue banks, right? So these are the practical considerations here. We want at least 100 samples of that type from your cohort, ideally 500 samples. No PBMCs, because that's well covered already. Primarily healthy. I think disease is admissible also. This is to be determined. But we've got to start with healthy. And GTEx was a healthy dead cohort, if there's healthy dead. We'll need genetic data, so whole genome sequencing, exome sequencing, genotyping chips, and so on. And of course, we'll need metadata to be as rich as possible as in any human cell atlas project. So what can HCA Asia do, right? Let's come up with some ideas today before you all leave. Thank you.
Partha.
A quick question, Shyam. Are you proposing to do single-cell DNA sequencing and single-cell RNA sequencing, or is it bulk DNA sequencing and single-cell RNA sequencing?
I was suggesting bulk DNA sequencing and single-cell RNA sequencing, but if there's single-cell DNA sequencing would be very interesting. Also, slightly different objectives, perhaps more basic. Yeah.
Bothia.
Really nice direction to go. So in terms of the design of the project, do you think it's better to do one cohort, multiple tissue, or is that too difficult? We have to use multiple different sample sources from different groups. And what are the advantages and disadvantages of each sort of collection strategy?
So I think it's an excellent question, but I'm going to give a very short answer. The decision has already been made. It's a small number of tissue types, but very diverse cohort all over the world. But which tissue types is not set in stone yet, I think.
Thank you.
Great. Yeah, really great initiative. So since we have a limitation essentially on the number of samples that we can do because of the challenges with the pricing, is there some thought around actually choosing based on the genetics or the availability of people with homozygous variants so that you can get slightly cleaner EQTLs eventually because of the genetic diversity which we represent in Asia? That's actually possible, where we have a discovery stage where people are genotyped. They're, if you have a homozygous or a heterozygous person, and we post after the genotyping stage, we actually select who do we want to actually take the tissue from, if that person, if that tissue is available.
That's a very interesting question. There's a number of cohort selection strategies. What you're suggesting is fairly sophisticated. I think GTEx was fairly unbiased, and so my guess is this will also be unbiased because there are so many other constraints on setting up a project like this. If you add one more requirement that the cohort has to be genetically interesting, that may make it harder for some people, but of course, if you have a cohort, then you have the wherewithal to make it genetically interesting. That would be fantastic, right? But it's not a requirement.
Okay, maybe next quick question, please.
So I have a question about the opposite set of confounders, which would be the environment. So if you're aiming for 100 to 500 samples, what's the trade-off between keeping it diverse regionally, but also not drowning in batch effects and environmental covariance?
Yeah, so batch effects is the killer for any flagship project we propose here. In the GTEx context, there's a slight escape route, meaning each individual cohort should be at least 100 to 500 samples collected without batch effects within that cohort. And then, it's an Indonesian cohort, an Australian cohort, or a Singapore cohort, and a European cohort. They'll have their respective batch effects for sure. But then in the genetics community, that's quite common, GWAS integration across cohorts. So there are methodology for that, as long as each individual cohort within itself has minimal batch effects, right?
All right. Okay. Thank you, Shyam, very much. So I have to apologize, but for the interest of time, we will not have the panel discussion now. But at the end of today's session, Kristen Wells will invite all of you and the speakers back, and we'll have a more intense panel discussion, and potentially these flagship projects can be discussed then. So let's give one big round of applause to all the speakers again. Yeah. Just one quick takeaway that I got from today is that there's a lot of opportunity for cross-hybridization. I think these 5 projects that have been pitched don't necessarily have to be 5 independent projects. I think there are opportunities where they can really overlap and be synergistic. And I really encourage all of you, now we're going to have a poster session, but talk to these speakers and reach out to them. And really, as Sean mentioned, start writing proposals and see where it can take you. So I really encourage you to do this. Also, I just want to mention quickly that Human Cell Atlas Asia has a regional office. It's both in Japan and Weekend, led by Yoshi Ando, who's coordinating this meeting. But we're also starting an office in Singapore in GIS. And for that, Human Cell Atlas has generously provided opportunity to recruit a coordinator. So this person's role would involve coordinating such type of flagship projects, helping you to get the right information and right connections that you need, and also help to organize meetings like this and workshops that we plan to organize in the future. So position is open. There's an available spot. It will be in Singapore office. So I would welcome any of you who know anyone in your network who wants to take up on this exciting role of a coordinating role for Human Cell Atlas Asia. Please let me or Sham know, and we'll be happy to connect them to the right procedures.